hormone, say a thyroid hormone rises in blood, the signal is going first to the pituitary, then to the hypothalamus, so that the stimulating factor, that releasing factor, or that is coming down, or maybe in some cases there are inhibitory factors which are now inhibiting that pituitary hormone, which are the tropic hormones. What is tropic? What do you mean by tropic? Term tropic. Tropic, it is mainly, this term tropic is mainly used in relation to endocrinology, endocrine physiology. Tropic means something which is secreted from one gland and influencing another gland, which he was telling at the beginning. Hormone definition does not mean that it will target another gland only, but tropic means secreted from one gland. So, tropic hormones are those secreted by the hypothalamus and those by the pituitary. Right, because they are going and acting on another target gland. For the hypothalamus hormones, they are acting on the pituitary gland. Most of the hormones of the hypothalamus, they are acting on the pituitary gland to most. Most why? Because hormones, some two of them are not acting on the pituitary gland to, that we will discuss in today's class, to secrete some other hormone. But most of them, like say CRH, releasing hormones, they are acting on the pituitary gland to make the pituitary gland secrete something else, some other hormone. So, that is tropic to the pituitary, and the hormones of the anterior pituitary are now tropic to the target glands like adrenal, thyroid. Okay. Now, basic anatomy of the hypothalamus, which you will learn later when you do the head, neck, brain part in anatomy. Okay, so basic hypothalamus is divided into various nuclei and various areas. Now, these names are somewhat not misleading, I would say, but they have been named such and you have to remember as such. If not in physiology in detail, but when you will learn about anatomy, you have to remember. See, dorsal means usually we mean posterior only. Ventral means anterior, but see, dorsal hypothalamic area, but posterior hypothalamic nucleus. You may ask your anatomy teachers why such naming has been done. Even I don't know, but posterior hypothalamic nucleus, say, see, anterior hypothalamic area, again ventromedial nucleus. Now, ventral and anterior, commonly we know in medical terms, it's almost the same thing. Our anterior portion is the ventral, posterior is the dorsal, like this. So, dorsal becomes posterior. What is dorsal? When we crawl on our knees at the beginning of life, so what is dorsarial later becomes posterior, anterior is ventral. But here the namings are such, so various nuclei and various area, and here nucleus means something, again something else. So, nuclei are collection of cell bodies, we know that. So, it may be that anatomically, or it is anatomically, that there are various nervous systems in the nucleus. It is actually that cell bodies are collected. So, inside the central nervous nucleus in biology means two things, usually designate two things. One is the cellular nucleus, the nucleus of the cell, maybe a plant cell or an animal cell, there is a nucleus obviously, but except there are exceptions. There are exceptions. Tell one exception. Very good answer. Mature mammalian RBC. So, only RBC. So, mature mammalian RBC. Up to which stage RBC does not contain a nucleus? Which stage of erythropoiesis? Or from the question is which stage? These are MCQs also. From which stage the RBC nucleus is disappearing? So, erythropoiesis is a very common question from hematology for your exam purpose and of course for knowledge purpose, erythropoiesis. So, there is a stage where the nucleus, so that is another topic. So, you have already been taught, so you should be able to tell. So, nucleus, but in central nervous system, nucleus means nervous system, nucleus means in collection of cell bodies inside the central nervous system in nervous system, and ganglion is a collection of cell bodies or ganglia in plural outside the central nervous system. Now, these are nuclei. Here there are collection of various cell bodies of neurons, cell bodies of various neurons. They have names, most names we do not require as of now. So, when you will be learning this in anatomy. So, where is the hypothalamus? Before that, where is the hypothalamus situated? It is situated in the floor of the third ventricle. in the lower part of the brain, okay, below the cortex obviously, and also the thalamus. So the name hypothalamus, but just above the pituitary gland. So in the obviously in the as a part of the central nervous system, and hypothalamus is a very important organ. So hypothalamus, so that is the location of the hypothalamus. Now basic anatomy, various nuclei and various areas, and what is labeled as optic chiasma we will learn later in the sense when we discuss towards the end of the year during the special senses. Optic chiasma is related to the carriage of the optic visual signals by the optic nerve to the brain, optic nerve to the brain, from the eyeballs to the brain. So optic chiasma is just labeled for its location there. It has nothing to do as such with the hypothalamus and its functions. Median eminence is a part of the hypothalamus from which most of the hormones are secreted, especially which are those hormones which are influencing the anterior pituitary. And hypophyseal artery, hypophysis is another name for the pituitary gland. These arteries, whatever you find hypophysis related terms, those are the related terms to a hypophysis. Pituitary gland is also called hypophysis. Anterior pituitary is called adenohypophysis and posterior pituitary is called neurohypophysis. Now why this naming is also not very not corresponding to each other? What does the term adeno mean? Adeno means, anybody related to? Adeno is related to glands. Now pituitary is itself a gland. Now I don't know why the anterior pituitary is separately called or just the anterior one is called adenohypophysis, because adeno does not means anterior as far as I searched out even through Google searches and all, and adeno, nowhere it is written adeno means anterior, but pituitary hypophysis is already pituitary, adeno is gland. So anterior pituitary gland is adenohypophysis, but neurohypophysis is posterior pituitary. Posterior pituitary usually is a collection, contains a lot of axons whose cell bodies are in the hypothalamus. So there is a lot of neural tissue in the obviously there will be vascular tissue, otherwise how will a organ get its oxygen supply and also but there is a lot of neural tissue, and probably that's why posterior pituitary is called neurohypophysis. But that does not mean anterior pituitary is more vascular, but adeno is not related to this vascularity. It is adeno itself means related to glands. Adenohypophysis is the pituitary gland, but the anterior part of the pituitary gland. If it was used in the strict sense, then adenohypophysis should mean the whole gland, not only hypophysis. But adeno is related to gland, somehow it is named. And the posterior, see the pituitary lobes are, see here, okay, you see the anterior lobe. See the anterior lobe in the diagram, the pointer is, it is mentioned, but that does not mean, it is shown in the diagram, that does not mean the posterior pituitary is devoid of blood vessels, then it can't survive. But there are more of neural tissue in the posterior pituitary. So here no blood vessels are shown, but that does not mean it is devoid of blood vessels, then it can't survive. But there are a lot of neural tissue here, and it is called the neurohypophysis. Now this is the same thing related to the anoid. See, it is situated in the sphenoid bone is labeled here, sphenoid bone. So pituitary will discuss next class. It is located in the sella turcica, one cavity is there over in the sphenoid bone. So that is labeled here. So again the lobes are shown, okay. The median eminence, this is a separate, that is a different kind of section where the median eminence, it is shown from the front, that is shown from the side. Median eminence is shown. Hypothalamus is situated in that cavity of the third ventricle. Here also the optic chiasma, these are all labeled. See, sella turcica is also labeled, pituitary fossa, all these things are labeled. So this is the basic anatomy. Here you can see again the most of the blood vessels are shown in this is in the anterior lobe of pituitary, and here the posterior pituitary is nothing is shown, but that does not mean nothing is there. See, here it is labeled. Nerve tissues are coming down. There are a lot of axons of nerves in the posterior pituitary, and also there are blood vessels. See, from the in the posterior lobe of the pituitary, the hormones which are being released from the posterior pituitary, they are going out through the blood vessels of the, here the blood vessels of the posterior pituitary is shown, here the anterior lobe. In this diagram nothing is shown. So just for avoiding congestion in the diagrams, they are showing in separate diagrams, but that does not mean that the posterior pituitary is devoid of blood vessels, or here the anterior pituitary is devoid, not like that. So the diagrams are just representative, they are shown for clarity, shown like this for clarity. So this is the basic, anatomy. Also see here it is somewhat congested, so many things labeled in same diagram, but here the labelings are, that means more things are written. If you look at this diagram, no need to just look at, no need to take pictures or something. These are from standard textbooks of physiology. See, here it is written, large body neurons make large body neurons. Okay, we will discuss the hypothalamic posterior pituitary hormones, we will discuss this. Now, basic functions of hypothalamus. Hypothalamus does not have only endocrine function. So, basic functions of hypothalamus is a separate class. We will do with the central nervous system. And in fact, temperature regulation may itself take one half of one class or more. Okay. So, basic functions of hypothalamus, just look at this thing and just remember the points, but for now we will have separate classes on that. Hypothalamus, temperature regulation is done by the hypothalamus. So there are integrating areas, just afferents you forget for now, there are afferents. In fact, temperature is a sensation, just a general sensation. Afferents will come from all parts of the body, from skin, from all parts of the body, etc. And there will be centers in the deep tissues, etc. So for now forget the afferents. There are different integrating areas in the just you had a look at the nuclei and the areas. Integrating areas for different hypothalamic functions are different. So same area may have more than one function, but they have different. So say for temperature regulation, anterior hypothalamus gives a response to heat, posterior to cold, and temperature regulation will be dealt with, and the hypothalamus will be dealt with in a separate class. Just for now, just I am mentioning the functions. Temperature regulation is one of the functions. Neuroendocrine control, what we are concerned with right now, neuroendocrine control, this function of the hypothalamus. Appetitive behavior is thirst, hunger and sexual behavior. These three are called, classified in appetitive behavior. Thirst, hunger and sexual behavior, these are appetitive behaviors. These controlled by the hypothalamus. Again, there are different areas. Neuroendocrine function also different areas, we are coming to it. And defensive reactions, that is, defensive reactions include fear and anger. Okay. So, somebody is maybe busy with mobile over there, and my defensive reactions, if they are now, if I am emotionally now take that too emotionally, my hypothalamus will work, and this work together with the limbic system. Hypothalamus works together with the limbic system for emotional responses. That is also here they have written it as defensive reaction. And body rhythms, mainly sleep rhythm. So, if you are serious in attending classes at 8 AM from the next year, now 9 AM, if you are serious enough to attend every day, you should be up at least by 7:30, not later than that, maximum 8. Okay. So, now if you have a habit, so I will do whatever, study or do anything else as per my wish up to 2 AM and get up at 8 AM. This is your routine, or 1 AM, 8 AM, 1 AM, 7 AM. And if you maintain this for say 15 days or so, the body, the hypothalamus will recognize it, right? And now you don't need an alarm clock anymore. But one day you see friends together see a film up to 4 AM, next day it will not work. May not work. Still it may work if you think, no, I have to attend the class or do something else at 8 AM sharp. So still it may work, but it is probably, it may or may not work. But if it is a rhythm and you are maintaining it for a long period of time, this rhythm will work. So that is done by the suprachiasmatic nucleus of the hypothalamus. So two sides, two nuclei, suprachiasmatic nuclei of the hypothalamus, and afferents are from the retina. Okay. So all body rhythms, they have written body rhythms, but it is the sleep rhythm. All these charts and etc. are from standard textbooks. I don't think you will find it in the books, but no need to take pictures or something like that. The information, all this information you will get in your books, which the books you follow, but just have a look at them. So these are the various functions of the hypothalamus, and today we are concerned with the neuroendocrine control. Now why neuro? In some cases, see, you just can see growth hormone, prolactin, FSH, LH, ACTH, TSH. With ACTH you also mentioned beta LPH. These are hormones of the anterior pituitary. So those are under vascular control. That means from the hypothalamus, hormones are secreted which are going to influence the pituitary to secrete these anterior pituitary hormones. But for the vasopressin, oxytocin and catecholamines are secreted from the medulla of the adrenal gland. Adrenal gland has two parts, cortex and medulla, outer cortex, inner medulla. For two organs at least, we distinguish cortex and medulla very clearly, that is kidney and the adrenal gland, maybe other organs also, but kidney and the adrenal gland. In basic our discussion of physiology, we read about the cortex and medulla separately and the functions. So adrenal cortex, adrenal medulla, from the adrenal medulla, catecholamines, namely epinephrine and non-epinephrine, also called adrenaline and noradrenaline respectively, are secreted under the influence of the hypothalamus and direct neural influence. Same for vasopressin and oxytocin of the posterior pituitary, but for the others, these corticotropic hormones, tropic means pituitary will again influence another gland, that is tropic to some other target gland. Those tropic hormones of the pituitary are influenced by some tropic hormones from the hypothalamus which are coming to the anterior pituitary through blood vessels. Now this is somewhat it was I think it should have been done in the introductory class. So this is just the hierarchical control, hypothalamus controlling the anterior pituitary. Again here they have given the example of an adrenal cortex hormone, cortisol. Cortisol is one of the hormones of the adrenal cortex. So glucocorticoid, cortisols, we will read about adrenal cortex in detail in the coming classes. That means endocrine physiology will take a lot of classes, maybe 2 months or so. So adrenal cortex will be dealt with separately, adrenal medulla, all will deal with. So see, anterior pituitary, ACTH is coming to the adrenal cortex, cortisol is secreted, then cortisol is going to its target tissue. And hierarchical control means just grandfather, father, son, like this hierarchical control. Hypothalamus itself has some, its integrating area is again in the cerebral cortex. Just a simple thing, just the hierarchical control and feedback. How is the feedback organ? One hormone from one organ, one target organ like pituitary, then another hormone from there, another target organ, but that target organ is sensing. That means if, say for example, thyroid hormone is getting decreased in blood, then thyroid is sending back the signal to the pituitary, and then again through that is feedback, and this is called a negative feedback. Positive feedback, negative feedback, I think you should know by now, general physiology class. At the first, homeostasis, balance of the internal environment of the body, this was done. Homeostasis. So positive feedback, example of positive feedback, but most feedbacks in the body are negative feedbacks. All these have been done. Homeostasis was the first class. At the beginning of the year, those who are new, you have to read it by yourself for now, and if you feel any problem, you have to come to us, all those topics which are covered. Homeostasis is in the beginning of the year. So what is positive feedback, what is negative feedback? Examples in endocrine physiology, most are negative feedbacks, except maybe 1 or 2. Okay, so those will be done in due course of time, we will learn. So endocrine, mostly negative feedback. So this is simple feedback loop. This is more of an introductory topic on endocrine. Again you see, when your peripheral gland means like the thyroid or the adrenal, it is also labeled, also the gonads. So gonads are the testes in the male and the ovaries in the female. Gonads, liver also, okay, insulin, glucagon, liver. So whenever a hormone, the endocrine gland is not, what makes you laugh? Whenever an endocrine gland is not working properly and the hormones, a thyroid hormone is gland is failing, and T3 T4 is not being produced in enough amount. So that is called hypothyroidism and primary hypothyroidism, primary endocrine disease. Whenever the issue is at the level of the pituitary, it is secondary. So thyroid gland is okay, but pituitary is not functioning properly. So pituitary can't secrete TSH, so thyroid can't secrete T3 T4, but thyroid gland is itself okay, then it is secondary hypothyroidism. And if it is at the level of hypothalamus, it is tertiary, but that tertiary term is very rarely used. When you will be, some of you may become medicine specialist and then endocrinologist. Mainly these two terms are more commonly in your medical jargons. You will have to diagnose these things, whether it is primary or it is secondary. Tertiary, sometimes it may be required, but not in most cases. So if the hypothalamus, the TRH, thyrotropin releasing hormone, is not functioning, and for that reason the pituitary is okay and the thyroid gland is okay, or the adrenal is okay, pituitary is okay, but there is corticotropin releasing hormone is deficient, then that is a example of tertiary endocrine disease. So that is what is written here, mentioned here. So got it? These are simple things, I think. At your level, these should be simple enough. Not tertiary, secondary, primary, these may be new to you, but that there may be deficiency in functioning of the hypothalamus or the pituitary or the thyroid gland. Usually all are not deficient together. So that is a very rare situation. Everything is not working. So if the deficiency is at one level, then there may be some deficiency at some particular level. That concept should be simple enough. Now the terms primary, secondary, tertiary may be new to you. So that's why it is being told. Right. So primary endocrine disease is what is at the level of the peripheral gland. Secondary endocrine disease, but when the medicine, you will attend the medicine theory classes, they will expect that you already know these things from your physiology classes, right? So they will not go into the details of these things. They will just start a chapter and mention this thing, primary hypothyroidism. So you should pick it up. Primary hypothyroidism means that the issue is at the level of the thyroid gland. They will expect, medicine teachers will expect, or that you already know these things from your physiology concepts in physiology. Now see the anterior pituitary hormones, which we will be just discussing next day. So what are the hormones from the anterior pituitary? See the last line. ACTH and along with beta LPH, beta lipoprotein, protein TSH, FSH and LH, okay, together called the gonadotropins, growth hormone, prolactin from the anterior pituitary, and these are being the hormones of the hypothalamus which are influencing these secretions of these hormones. For the ACTH and beta LPH it's CRH, corticotropin releasing hormone. For TSH it's TRH. For LH and FSH it's GnRH. For growth hormone it's GRH and GIH. GIH means GIH, it is no longer called GIH, growth inhibiting hormone, it is called somatostatin. Somatostatin you have heard also in the pancreas. So these some chemicals are there which are released from some other areas also. But somatostatin, this earlier it was called GIH, now it has been seen it is same as that somatostatin, chemical nature, research has shown it is same as that somatostatin. There it is subserving some other function in the GI tract. Here it is somatostatin is growth inhibiting hormone, that is it will inhibit the pituitary from secreting, anterior pituitary from secreting growth hormone. Again PRH and PIH, prolactin releasing and prolactin inhibiting, but PRH is somewhat dubious till now. Okay, PRH has not been identified, physiologic PRH chemically has still not been identified properly. So I think you better omit. If you are asked to write the names of the anterior pituitary hormones, which we will discuss next day of course, You write 1, 2, 3, 4, the hormones of the hypothalamus, hormones of the hypothalamus. 1, 2, 3, 4, 5, you write 6, not 7. PRH, I think you should not mention, it is still under research. A receptor, hormones obviously will act through receptors. A receptor has been identified in the pituitary, which is believed to be acting receptor for PRH, but no such chemical. One 31 amino acid chemical has been identified from the hypothalamus, which may be acting like PRH, but whether it is actually PRH is not, it's still under a lot of research is going on. So the standard textbooks of physiology are writing the same, that research is going on. So I think at MBBS level you don't mention PRH. You here on prolactin you mention mainly inhibitory role of the hypothalamus. Prolactin has a influencing role, that means prolactin, so you will ask, so if prolactin decreases, what will increase? Prolactin, not TSH of course, TRH. TRH has an stimulating action on prolactin secretion. But don't say about PRH. If not you are asked separately, if the examiner in the Viva exam, maybe if the examiner asks you about PRH by himself or herself, then you may mention that it is under research or something. But if it is asked who stimulates prolactin secretion, first you tell TSH. Now see, by now already you also know the actions of the hypothalamic hormones, these hypothalamic hormones. CRH will have stimulatory action obviously on beta LPH and ACTH, because CRH is corticotropin releasing hormone, not inhibiting. TRH is, most are stimulating, hormones are stimulating. TRH is on TSH and also TRH stimulates TSH and also prolactin. GnRH earlier it was called LHRH, and the people thought, that means the scientists thought, that there is a separate FSHRH which was not being identified for a long time. It was called luteinizing hormone releasing hormone, but later it is found the same also stimulates FSH secretion. So now they call it GnRH. They are stimulating both LH and FSH. Now with ACTH we speak of beta LPH, but beta LPH still now it is a separate chemical, no doubt. Chemical nature has been identified, but physiologic role has not been identified. So if you are asked to name the pituitary, anterior pituitary hormones, which we will do next day, then you mention this at the last, beta LPH. ACTH, TSH, LH, FSH, growth hormone, prolactin, no problem, 6 of them. In the 7th, you mention beta LPH, and in case of the hypothalamic hormones which are stimulating or inhibiting the anterior pituitary, you don't at all, you omit PRH. That's my suggestion, you omit PRH unless you are asked in some MCQ or in the Viva. Some MCQ say it is hinting particularly at PRH, then only you should mark it. But otherwise, you should avoid mentioning PRH, and for beta LPH in an anterior pituitary, mention it at the last, at the 7th of all the 7. Right? So, and from obviously through blood vessels, all these are hypothalamus. These releasing and inhibiting factors were earlier called such, releasing inhibiting factors. But now we know they are coming through the blood. These are tropic hormones. Tropic means influencing another gland, pituitary gland. These are tropic hormones influencing another gland. So this is coming through the blood, that means route is blood through the anterior pituitary, and then influencing the secretion of these tropic hormones of the pituitary gland. Now the CRH, TRH, GnRH, these are called hypophysiotropic hormones. That means tropic to the hypophysis, tropic to the pituitary, influencing the pituitary. These terms should be known to you. Hypophysiotropic. And pituitary hormone is adrenocorticotropic. That means tropic to the adrenal cortex. Thyroid stimulating hormone is also called thyrocorticotropic. That is tropic to the thyroid gland. But hormones of the hypothalamus are tropic to the pituitary, hence also called hypophysiotropic. HYPO, then hypophysis, PYHSIO, hypophysio, TROPIC. Hypophysiotropic, not trophic, hypophysiotropic hormones, because they are tropic to the pituitary. Hypothalamic hormones are hypophysiotropic. That means hypothalamic hormones which are influencing, stimulating or inhibiting the anterior pituitary are the hypophysiotropic hormones. And from the earlier chart you see, they are secreted from different areas. So they may be asked in MCQs. See, prolactin via PRH. PRH you don't mention usually. PIH is from the arcuate nucleus. Okay? All these you will get in your books. Say SCT, CRH is from the paraventricular nuclei. FSA, GnRH is from preoptic area, especially its medial part, medial preoptic area. And TRH and CRH all paraventricular nuclei and neighboring areas. TRH and CRH from paraventricular nuclei and neighboring areas. GnRH from medial preoptic area, you add the word medial here. PRH you leave it for now. PIH from arcuate nucleus, but PRH, if it is mentioned in an MCQ, PRH is supposed to be secreted from, like this MCQ. In the MCQ you find the term arcuate nucleus, you mark that. Okay? And GRH, growth hormone, from the periventricular and arcuate both. Now periventricular and para are separate things. Here it is peri, there it is para. So these may be potential MCQs. Or if you get arcuate nucleus secretes the following hormones, in the option, if you don't get GRH, you don't get PIH, you get only PRH, then you mark PRH. Have you got this thing? PRH you try to avoid in discussions, but you get an MCQ where it is written arcuate nucleus secretes, and you don't get PIH or GRH in the option. Growth hormone, releasing hormone, you don't get. Also somatostatin, GIH, somatostatin, you don't get any of these. Usually that is a very bad MCQ to frame. PRH is obviously dubious, and why you will frame MCQs like, why the examiner should frame. But if someone frames where somatostatin, GRH, PIH, nothing is available, arcuate nucleus secretes, then you go for PRH. Or PRH is secreted by, in the option there is arcuate nucleus. Also it is written other areas, but arcuate nucleus, then you mark arcuate nucleus. So this is pretty simple. Now vasopressin and oxytocin are called hormones of the posterior pituitary. Why? From the posterior pituitary it will go out through blood vessels and act on the vasopressin mainly on the kidney, in the kidney. How and where, it is renal physiology discussion. Oxytocin on the breast and the mammary gland, breast and mammary glands are the same thing. Breast and the uterus, uterus during childbirth, breast during milk ejection reflex. Oxytocin, vasopressin function we'll do in the next class. But oxytocin, vasopressin are hormones of the posterior pituitary and not of the hypothalamus. Why not of the hypothalamus? If they were hormones of the hypothalamus, by definition of hormone, they should have come through blood vessels. Understand this thing. Oxytocin or vasopressin are secreted from the hypothalamus, but they are hormones of the posterior pituitary. Potential explain why question. Explain why type of question will be, there will be 4 questions in each paper. Hormones are vasopressin and oxytocin. Vasopressin is also called arginine vasopressin in the human and most mammals because it contains arginine. In some mammals, it is replaced by lysine. Arginine is replaced by lysine. There it is called lysine vasopressin. In the human it is and most mammals it is arginine vasopressin, also labeled as AVP, oxytocin OT, arginine vasopressin AVP. It is secreted, see, hypothalamus, synthesized in the hypothalamus, released into the posterior pituitary, target of then it goes to the collecting ducts of kidneys and from oxytocin on the uterus and the breast. But these are called hormones of the posterior pituitary. Why? Because by definition of hormone should travel through the vascular streamline, not through neural streamline. It is going from the posterior pituitary via blood to the target organs, collecting duct, uterus and breast. Functions we will discuss in next class. And but precursors of these hormones, actually these hormones in proper are not secreted from the hypothalamus, just precursors are synthesized in the hypothalamus. Precursor for vasopressin is pre-pro-vasopressin. Pre-pro-vasopressin, precursor for oxytocin is pre-pro-oxytocin. These are synthesized in the hypothalamus, and hypothalamus and pituitary has, posterior pituitary don't have vascular connections, they have neural connections. Posterior pituitary vascular supply is something else. You will read in anatomy, study in anatomy, something else, even I don't remember now. So see, oxytocin, vasopressin from the hypothalamus, but they are coming down via these neural connections. These neural connections are carrying pre-pro-vasopressin and pre-pro-oxytocin towards the posterior pituitary. And now in course of this carriage, course of this transport, this pre-pro-vasopressin, there is something called neurophysin. Neurophysin means, which is usually they were thought to be binding proteins for oxytocin, vasopressin, neurophycins, but now they are saying that neurophycins are part of the precursor molecule itself, breaking down. So pre-pro-vasopressin is breaking down into neurophysin 2 and pre-pro-oxytocin into neurophysin 1. Pre-pro-vasopressin into neurophysin 2. It is called neurophysin 2 and vasopressin. Pre-pro-vasopressin is breaking down into neurophysin 2 and vasopressin. Chemical nature are separate for all these chemicals. And pre-pro-oxytocin is breaking down into neurophysin 1 and oxytocin. And now vasopressin are during carriage towards the posterior pituitary, during transport, now getting stored in the posterior pituitary for some time and then released into the bloodstream to act on target organs. For oxytocin, it is the uterus and the breast, and for the vasopressin for the in the kidney. Okay. So these are hormones of the posterior pituitary, but precursors are synthesized in the hypothalamus, coming down through neural connections. Okay, in anterior hormones of the hypothalamus secreting the anterior pituitary, I mentioned to forget one thing. For CRH, a binding protein has been identified, CRH, which binds CRH and inhibits its action. So you may ask, if ACTH has to be secreted in less amount due to some, say cortisol has, adrenal cortex hormone has gone up in blood, ACTH has to be inhibited. Now there is no CIH in the diagram, so how is ACTH inhibited? There is a CRH binding protein and there is no CIH as such, but CRH binding protein in peripheral blood which binds CRH and inhibits its action. Got it? Which binds CRH and inhibits its action. Peripheral blood also in the central nervous system, CRH is binding, but no such binding protein for TRH has been, thyroid releasing hormone, thyrotropin releasing hormone has been identified. So thyrotropin releasing hormone, what is thyrotropin? Thyroid stimulating hormone is also called thyrotropic hormone or thyrotropin. Now thyrotropin releasing hormone and CRH is corticotropin releasing hormone. For CRH a binding protein has been identified, but not for other hypothalamic GnRH. Now how those are inhibited, that means how LH, FSH, TSH are inhibited are still under research. But how ACTH, beta LPH is inhibited, it is by binding of CRH to the CRH binding protein. Okay. So time is also over, this you don't need for now. Those targets you don't need for now. So this is about the hypothalamus and the how it is acting on the pituitary. Hypothalamus to anterior pituitary is vascular connection, posterior pituitary is neural connection, and the hormones we have named and the areas. CRH is from paraventricular nuclei, TRH also, GnRH from, which is from medial perioptic area, GH from periventricular nuclei, and PIH from arcuate nucleus, you try to avoid TRH, but if you are compelled to mention it, then it is from the arcuate nucleus. And also potential MCQs explain why hormones vasopressin, oxytocin, although their precursors are synthesized in the hypothalamus, these are hormones of the posterior pituitary. Next day we will go on to the hormones of the pituitary. Especially growth hormone is a separate discussion that will require another class next Saturday. Okay, thank you.
Okay, good morning everyone. Anyways, before life we have this with embryological development. We are continuing. We are still in the general phase of embryonic development, which is kind of complex story involves in this particular process. Moreover, is invisible, just like any other parts of the human biological process is, but this is a series of the thing which keeps on happening like stage by stage. So again, to cut down this particular complexity, we just like learn this particular embryological development in phases, like in terms of the weeks, how in a week it develops. So far our embryo, we have reached to 2 weeks of life. So it has reached to this particular 2 weeks of life. Before that we have this fertilization, and from this particular 2 weeks of life we have achieved some parts of this particular contents or some parts of this particular developmental things, which is this. So it is by the end of 2 weeks life, this embryo, which was initially a single cell, took several divisions, took several modifications, and by the end of 2 weeks it is planted well enough. It is housed inside this particular uterus. We call them as endometrium. Now we call them as a decidua because of the changes which is happening there. And this particular embryo have got cavities inside. We call them as yolk sac cavity and amniotic cavity. It has 2 layers of the cells. We call them as bilaminar germ disc. The 2 cells are different. We call them as epiblast cells and hypoblast cells. And it has hanging over these particular connections between it. We call them as connective stalk, which is present holding this. And it is floating through, and gradually it is anchoring deep into this endometrium in the south for getting nutrition, oxygen, etc., etc. So it can grow big. Further development can be housed, and further development can be sponsored. Now, from these 2 weeks, we are moving into 3 weeks. So 3 weeks itself is a complex phenomenon. Of development goes on, and 3 weeks is, for the long time we see that is a very hot topic for the embryological development as far as exam questions is concerned. We see that 3rd week or entire 3rd week development is coming as a exam long questions, or maybe a fractions of this particular 3rd week development comes as a short notes as well. Likewise, the 2nd week, like entire phenomenon, what happened in the 2nd week also is an exam question or exam long question. But more popularly, we see that the 3rd week development of the embryo is coming as a exam long questions or short questions, more commonly, more popularly. So in this particular phenomenon, 3rd week, we are encountering a series of development in this embryo. We divide into 2 different halves. So a part of the development which goes on this particular process, we give a name to it. We call them as gastrulation. We call them as gastrulation, which involves 2 phenomena, subphenomena. And other process into this particular 3rd week, which remarkably happens of this embryonic phase of life, we call them as neural tube formation. That means the earliest form of brain tube formation that also happens or beginning to happen in this particular 3rd week. So in the 3rd week, we have 2 phenomena to address, and within this particular 2 phenomena, we call them as gastrulation and neural tube formation. In terms, we are going to talk about these 2 entities and how these things are forming and how these things are coming. Now, taking in terms about gastrulation, as I am saying, this is a big process. The gastrulations can be divided into 2 parts. A part of the gastrulation is that by now 2 weeks, we have 2 cell laying or 2 cell layer. But as we are grown human being, we have 3 different cell lines. Now, we have to replicate 1 more layer from that particular 2 layer to make it 3 layer cells or 3 layer lines of these particular different things. So from 2 layer, 3 layer making or happening is going to occur in this particular 3rd week. This is a part 1 of gastrulation. And second phenomenon which is taking place out here, we call them as notochord formation. So, that particular notochord formation is actually triggering the further development, we call them as the neural tube formations. If you do not have the notochord formed, we do not have this particular neural tube formed. So, these are the phenomenon we are going through. So, for this particular session, first half we will be talking about the gastrulations, which is rather easy, I would say, composed of 2 elements. So, from 2 cell line we are going to acquire 3 cell line. So, bilaminar germ disc is going to become 3 layer germ disc or trilaminar germ disc, which you are going to have, and we have to have 3 layers because 3 layers generates 3 different types of the tissues in our body. Some part develops brain, some part develops liver, some part develops this particular gut tube, organs, etc., etc. That is why we need this particular 3-liner cell progeny and to develop on. And then we have this notochord, that particular phenomenon is required for formations of our brain, which happens to occur earliest on rest of the organ development. Yet, this particular organ, we am saying brain development happens to occur in the 3rd age or 3rd week of life. That starts functioning even beyond we are born. So, hardly we use the brain, right? Right now, how many of you are using brains? Very few, right? We are keeping it intact so that we can do something good about the brains, right? So, let us begin with the story. So, this is what we have so far, 2 weeks of life. We have 2 cavities, amniotic cavity, yolk sac cavity, which shrinks in size, becomes secondary yolk sac. Initially, we call them as primary yolk sac. And above one, we have this particular cavity, amniotic cavity, which is lined by a cell, which is known as this epiblast cells, and below one, we call them as hypoblast cells. And basically, they are developed on these particular areas and covering this entity. Now, we focus on this one cell line out here, we call them as epiblast line. So, this is what we calling here is the beginning phase of this 3rd week, we call them as gastrulation. There are 2 phenomena which is taking place. 3 layer cells we have to make, and other thing is the formations of the notochord that is going to happen. Now, this is what we are starting with. One line which are highlighting out here, which is red over here, is that epiblast cells. Now, this particular epiblast cells from this particular single layer is basically transforming into 3 different layers. So, from here we are going to achieve this particular double decker sandwich or whatever it is, 3 layers. We call them as ectoderm, then we have the mesoderm, then we have the endoderm. Now, this particular phenomenon we call them as gastrulation. That is what we are going to talk. This is an important, very short note, and you have to describe it as we are going to talk, right? Good. So, let us begin with it. So, this is a kind of like a three-dimensional thing which we are seeing here is this embryonic tube or embryo hanging with this yolk sac, with this particular yolk sac cavity and amniotic cavity and the disc in between. And this is hanging over here, we call them as connecting stalk. Remember that connecting stalk is going to become the future umbilical cord. Now, this particular region we call them as epiblast, this is known as hypoblast, and this disc we call them as bilaminar germ disc. Now, what happens in the bilaminar germ disc? Soon there appears a line in the midline within this particular disc. That particular line we call them as primitive streak. We call them as primitive streak. That line, a line appears in the midline. Now, as this particular line comes over here, this particular bilaminar germ disc is not yet divided on the poles, which is anterior pole, posterior pole, right pole, left pole is yet to be decided. Once this particular line appears, which is known as primitive streak, then we decide on the poles of the embryo. Am I clear? So, we have to divide over which end is the head end and which one is going to be the tail end. So, this particular line we call them as primitive streak. As it appears, that particular pole of the embryonic disc becomes the caudal end, that means tail end. Clear? Clear? As this line appears, primitive streak to this particular pole, that particular pole of this particular disc, this oval disc becomes caudal pole. The opposite one will be the cranial pole. That means, where you have this streak or the line appeared, we call this particular end as tail end of the embryo, and opposite side would be head end of the embryo. That is going to be the future head and that is going to be the future tail. We do not have tail, do we? Maybe surprisingly we may have hiding somewhere. So that particular pole we call them as the tail end of this embryo. Agreed? Now this is what we are calling here is primitive streak. What do you call them as? Primitive streak. What is primitive streak? The line appears. Now you see that within this particular pole, there is a one end embryonic disc have this line. Other end do not have the line. So that particular end is going to be cranial end. This is going to be our head, and this is going to be the caudal end. That means the coccyx will be in this side and this side. Now if this particular primitive streak appears in this region, so this side will be decided on will be the caudal end, and this side will be the cranial end. Am I clear? So once this particular streak is appeared, this particular streak grows and grows to the half length of the disc. It do not cover the entire length of the disc. Now which side is happening? This particular change is happening on the epiblast end or superior end towards this amniotic cavity end. It is not happening on the yolk sac end. Nothing change happening on this. So whatever the change you are seeing here is taking development on the amniotic side or epiblast area. So tissues or the cells on this region shows this particular linear streak appears on the beginning of the third week. We call them as primitive streak, and this head end of the primitive streak moves towards this particular region and becomes a little bit swollen, dilated. We call them as node. We call them as primitive node. So primitive streaks end up in a circular node. We call them as primitive node. Am I clear? Now this particular primitive streak, as it appears, it begins the beginning of the gastrulations. So before that we didn't have the gastrulation to begin with. Once the streak appears, we see this is the ringing bell. Now we have begin with the gastrulation phase. Third week have literally functionally have existed. All right, clear? Which side it is happening? Epiblast side. That means towards the amniotic cavity side you are seeing this change. On the bottom side we are not seeing any changes. So this is happening in the midline, and this particular strict midline thing creates the axis of the embryo, cranial end, caudal end, and right and left is decided and divided on this particular appearance of this primitive streak, which is happening here, and had a bulbous end. We call them as primitive node, which is heading down. Now this particular primitive node and a primitive streak is not encroaching towards the entire length of the disc. You see that this is the entire length of this particular disc. Now this particular primitive node and streak nearly achieves the half distance of the disc. So if this particular entire disc length is 5 centimeter, only near to this 2.5 centimeter, we see that midline streak is coming with the nodes. Rest is not occupied by this. So nearly half length is appears. This particular cells which is on the epiblast shows these particular changes. After this, we have two poles. Once is decided, one is on this particular region, which is this head region, and another central part which is coming over here like this region, which is on the caudal end. So these two ends or these two poles are given to the names. We call them as buccopharyngeal membrane end and this cloacal membrane end. So this particular poles, once this decided, appears to midline spots as buccopharyngeal membrane and cloacal membrane, and it stops at this particular point. No further development occurs in this particular membrane on this cloacal or buccopharyngeal. So this is what we are seeing here. This beginning of the third week, we have the linear band, primitive streak, which has this particular happening on this particular epiblast end, and it has a knot here. We call them as primitive node or primitive knot, and this is how this particular primitive node and primitive knots is present, and this nearly reaches to this particular half length of this. Now this particular primitive node, which is on the cranial end of this primitive streak, is there. Within this primitive node there appears a hole. That particular hole we call them as pit, primitive pit. Right. So functionally and finally we get this particular primitive streak, primitive node, and the hole inside we call them as primitive pit. So these are the sequel of the things that is happening into this particular phenomenon of the primitive node, primitive streak, and this particular primitive pit. Now as this time progresses, third week continues to grow, this particular streak becomes wide, becomes little bit dilated or more encroaching, and this particular streak becomes a groove. Then we call them as primitive groove. So this particular thing, as we are seeing here, little bit dilated, and front part is kind of expanded, which is holding like a node and a pit inside, and this particular thing, streak or line, becomes a little bit wide. Then we change the name from the streak to this particular thing as a primitive groove. So primitive groove, primitive node, and primitive pit. These are the formation that happened so far. Now, as these things are formed, the cells or epiblast cells that are present within this particular primitive groove or primitive node, this region starts to migrate. They start to migrate towards which direction? They step down, they go a little bit under. So, what underlayer to this particular epiblast we have? Under to epiblast, what cell layer we have? Hypoblast layer. So, it steps down and gets into or in between the epiblast and hypoblast and populate that particular space. So, this particular downward movement of the cells through this particular region, primitive groove, we call them as ingression of epiblast cells. So, same epiblast cells they start growing deep through this particular groove and house or populate themselves between this epiblast and hypoblast and populate this layer. So, it generates a third layer from this particular two layer, epiblast above, hypoblast below. We generate a third layer by this migrating cell from the epiblast. This cells, epiblast cells goes under to it and settles down here and populate there. We get the third layer out here. This third layer is developing within this particular embryonic disc. Now, this particular third layer we call them as intra-embryonic mesoderm. Compared to that extra-embryonic mesoderm, remember we talked about that in a second phase that happened outside the embryonic disc. We call them as embryonic mesoderm, which creates a cavity. We call them as extra-embryonic coelom, which splits into two layers, planktonic layers, planktonofluoric layer, which is happening in a terms of the membrane development. So, this particular third layer, which is getting inside and forming a purple layer, which is we call them as intra-embryonic mesoderm. Now, we get these three layers, one above, in the middle, and the bottom layer. Now, as this particular epiblast cells, which is going down and settling down in this region, this particular hypoblast cells in yellow gets transformed and creates the layer which is known as the endoderm layer. So, hypoblast slowly replaced by this particular same epiblast cells, replaced by and creates the layer, we call them as endoderm layer. Then we have this particular mesoderm, intraembryonic mesoderm, and then we are left with this particular epiblast. Then this particular epiblast layer transforms into ectoderm. So, this is the phenomenon which is taking place through the primitive stage. Cells goes down and move towards this particular regions and creates the third layer, which is known as intraembryonic mesoderm, and this particular hypoblast layer is replaced by it, makes this particular endoderm layer, which is the bottommost layer. Then we get the middle layer, mesoderm, intraembryonic mesoderm, and then the superior layer, which is lying on this, which is the epiblast layer, transforms into finally into ectoderm. So, that's how we get this particular three layer cells from this. So, we have this particular hypoblast replaced into this particular layer, we call them as endoderm. The middle layer we call them as mesoderm, and this top layer we call them as ectoderm. So, the phenomenon by which the cell goes deep inside, step down and populate this region, this phenomenon we call them as ingression of cells, ingression of epiblastic cell they are going through. And this particular ingression happening to different extent, all extent through this particular phenomenon. And this is how we create this particular three layers, ectoderm, mesoderm, and endoderm. So, ultimately all these cells which are coming here is coming from this particular epiblast or transformed epiblast. Epiblast cells itself gets inside, makes this mesoderm and replaces the hypoblast cells and makes this endoderm, and itself changes into ectoderm. So, we get this particular three layer cells now. Now, this particular three layer element which is known as trilaminar germ disc is achieved, which is a phase of gastrulation. So, this is what we started with. So, we had two layers, now we procure one more layer, now it is three layer. Now, this particular phenomenon of making from this particular two layer cells to the three layer cells, we call them as gastrulation. And how this particular gastrulation is happening? Because of this primitive streak, primitive groove, primitive pit, this forms and cell migrates down towards penetrating below. This particular penetration of the cells going below inside, we call them as ingression process. and getting inside, populating and creating this particular layer, endoderm, mesoderm and ectoderm. This is, we are getting here formations of all these 3 layers. So, this particular entire length of this particular embryo, which was bilaminar germ disc, now is trilaminar, except 2 spots. There are 2 spots which is still 2D, rest of this is 3D. So, 2D region is this region, which is known as this part, we call them as buccopharyngeal membrane, and this region we call them as cloacal membrane. These 2 spots remains to be 2D, that means only ectoderm and only endoderm is there. Rest of this particular disc is 3D, that means it has 3 layers. Alright. So, this particular region, buccopharyngeal membrane, is going to be the future mouth, and this particular region, cloacal membrane, is going to be the future anus region. So, these 2 regions are where we are not going to see the 3rd layer appearing. Rest of this particular embryonic disc have got 3 layers inside. Clear or not? So, this particular process we call them as gastrulation. This is what we have to narrate, this is what you have to write it step by step. How these 2 layers becomes 3 layers, what is the process which is happening. This is the phase 1 of gastrulation, which is involving bilaminar to trilaminar. This is one subdivision. Next stage of this particular phenomenon. Now, before we go into this next phase, now what happens to the primitive streak? That streak that appears in that particular thing, the functions of the appearance of the primitive streak is to produce this particular 3rd layer. Once this 3rd layer is produced, that means 3rd week off, 4th week onwards, this primitive streak should normally be absorbed or abolish or vanish. So, I repeat. So, 3rd week, this particular region, primitive streak, appears because we have to generate 3rd layer. Once this 3rd layer is produced by the end of the 4th week, we do not need any more primitive streak. That primitive streak should be job done and should just like close the office and shut off. But if it remains, it should not be remained. If it fails to regress or fails to disappear, it may create a disease. We call them as sacrococcygeal teratoma. It creates a tumor in this particular region of this sacrum coccyx region and remains like this. So, this is the remnant of this primitive streak. If it is not been completely absorbed, which normally it should, if it persisted as a cyst. We call this as a sacrococcygeal teratoma. Alright. So, this is a clinical aspects that you have to write about the sacrococcygeal teratoma, which is a remnant or persistence of primitive streak in the embryonic life, which should not be there. If it is present, creates a disease, we call them a sacrococcygeal teratoma. An embryo is born like this, the kids are born like with the tumor like this. So, we have to do surgeries if it is like coming out like this. So, this is what we have. So, now we have this particular 3 layers of this particular germ disc, 3-layer germ disc. Why you need that? As I am mentioning in the beginning, 3 layers of the germ disc produces 3 different parts of this particular body. Say for example, ectoderm produces the skin, the brain, etc., etc. The mesoderm produces the kidney, the reproductive system, etc., etc. So, endoderm produces the gut tube, etc., etc., liver, etc. So, these 3 different types of tissue, ectoderm, endoderm, mesoderm, produces different kinds of organ. So, coming back again, this particular germ disc is 3-layer germ disc now. We call them as trilaminar germ disc. This process of formation is, we call them as gastrulation, and this particular area of this germ disc is entirely 3D except 2 spots where you see that cloacal membrane and the buccopharyngeal membrane. These are the MCQs that commonly comes over here during this university exams. Now, the second phase of this particular gastrulation, which is notochord formation. So, formation of another thing we call them as notochord or notochordal process. This is a little bit complex than that what we have explained. So, previous one was the easy one, 2 to 3 is making not a difficult, just we have to add 1, create some layer, produce it and job done. This one little bit complex, but I am just not going to talk detail about this particular process, but just like talk with little bit in a simple form of formation of the notochord. Now, what is a notochord? It is a solid tube like structure that develops into this developing embryo during this process of gastrulation. So, it is a solid tube which is an embryological tissue that develops during the process of gastrulations, and this particular formations of this particular solid tube or notochord tube happens to occur sub epiblast layer. That means it is forming under the epiblast, like not on the top of it, it is forming under the epiblast layer. Now, what happens over here? We develop with this primitive node and we develop with this primitive pit. Now, this particular primitive pit which is ending somewhere in the middle of developing embryo. So, it is not approaching towards it. So, from that particular primitive pit, beyond that primitive pit, there is a development of further tissue which is growing towards this particular plate. We call them as procaudal plate, also known as this buccopharyngeal membrane. So, a tissue which is growing under the epiblast and growing towards this particular cranial end, towards this particular buccopharyngeal membrane side, we call them as procaudal plate, also known as pro or precaudal plate. So, pro or precaudal plate here, where the fusions of this ectoderm and endoderm is there, no penetration of mesoderm whatsoever happens there, which is going to yield this particular membrane side. So, this particular primitive node through which under this particular layer, yes, please. So, under this particular layer which is epiblast layer, this tissue which grows and grows towards this procaudal plate. Now, we coin this particular growing plate of the tissue as notochordal plate. Now, this particular notochordal plate starts develop from the primitive node and enters towards this and grows towards this procaudal plate. Now, soon after, the next phase of this particular notochordal plate happens. Within this particular notochordal process, the primitive pit, which was a small pit, was there, which grows into this particular procaudal plate and creates a canal. We call them as neuroenteric canal. So, this particular neuroenteric canal, formation of this little bit complex, but just like I don't go into detail understanding on. So, what happens here? We have learned about the primitive pit which was there in a primitive node. That particular primitive pit grows within this particular procaudal or notochordal process. So, this particular primitive pit, you see that this much of length was there. So, this particular primitive pit elongates within this particular notochordal process and grows big and grows anchors deep and creates a channel between this particular cavity above and cavity below. So, what cavity is lying on above? We call them as amniotic cavity, and what cavity lies below? The yolk sac cavity. So, this particular primitive pit now makes a channel between this amniotic cavity and this particular yolk sac cavity above and below, passing through this particular notochordal process and encoding over there and creating a channel or bypass. We call them as neuroenteric canal. So, it is creating a channel between it and making some kind of fluid flow through this particular thing. So, this is how schematically it looks like. This is this notochordal plate which is going in here, which is an orange arrow which is coming over here. So, this is the notochord which is developing from this particular primitive pit towards this particular buccopharyngeal membrane. We call them as procaudal plate, and within this particular notochordal plate or notochordal process, we have this particular pit which grows inside and creates a cavity. We call them as neuroenteric canal, and it creates some cavity between or tract between this amnion and yolk sac, right. So, formation of this particular channel, we call them as neuroenteric canal or neuroenteric canal circulation. Now, why this particular circulation is necessary, we do not have an exact clue on it. So, this is what we are talking about is notochord formations. Now, this is the phase 1, phase 2 of this particular notochord formation. Phase 1 is that we have this particular notochordal plate or notochordal process burrowing deep into this particular epiblast, growing towards this particular procaudal plate happens. Soon after, the second phase, the primitive pit which digs into this particular developing notochordal process and creates a channel. We call this as a neuroenteric canal, which basically creates a connections between this cavity above and cavity below, which is epiblast, which is holding the amniotic cavity with the bottom cavity, yolk sac cavity. And then we have this particular canal established, and later on this particular canal gets sealed, get absorbed. Now this particular canal should not be normally be present. So this particular neuroenteric canal should not be present, it should be sealed. If it fails to disappear, again it creates a cyst like sacrococcygeal teratoma. We call them as neuroenteric cysts. So that particular cyst again is a very complex cyst which develops into the neural system development. But again, so this particular entire phenomenon, as you are seeing with these formations of this particular channel, it should soon appears to regress, should not be present in between. So this is the second phase of this particular thing, and then there is a circular changes which happens within this particular notochordal plate, and it assembles to create a circular element which is solid rod kind of things is formed at this particular sub-epiblus layer and form a tube like thing which is making an axis of the embryo which is formed and which is known as notochord. So this particular notochord is formed from this particular primitive node to this particular buccopharyngeal membrane that side, and once this particular notochord is formed, this particular notochord gives the structural rigidity to this trilaminar germ disc and it creates the axis as well. So the upper axis like cranial end, caudal end, right and left, and this integrity of this trilaminar disc also been created. So this is the fate of these formations of this notochord, which includes the second phase of the gastrulation, which first phase 3 layer and then second layer is notochord formation. Now what happens to this particular notochord? The notochord have got two functions to come over here. Again, this particular notochord cannot remain there. So once this particular notochord forms, a part of the notochord is going to contribute the formation of further development of the embryo. We call them as neural tube formation. So the notochord actually triggers or stimulates or pokes to the formations of the brain tube. This is rule number 1. And second thing, notochord itself should be regressed. Only a part of the notochord remnant should be remained over in the midline vertebral column. So we have the vertebra, we have perhaps seen that vertebra. And in between the vertebra, which is the bones over there, between the vertebra, we have a disc, which is cartilage. Now that particular cartilage, we call them as intervertebral disc. Now that particular disc has a central part and has a marginal or peripheral part. The central part of this vertebral disc, we call them as nucleus pulposus. So that this notochord which is formed, which should normally be completely regressed, except a remnant of the notochord should be present on an intervertebral disc. That's a normal existence or residue of notochord that should be present. Anything else or anything beyond, if the notochord persists, creates some other problem, just like this sacrococcygeal teratoma. So this is the fate of this notochord. So it forms this particular, triggers the neural or neural tube formation. This is one. Second thing, it forms the nucleus pulposus. So what is nucleus pulposus? This is the disc we are referring to, which is lying between this vertebra. Now this disc has the peripheral part and has a central part. The central part, we call them as nucleus pulposus, which is a jello, which is a soft jello kind of thing, shock absorber. So this jello or nucleus pulposus is contributed by notochord, which is the normal adult derivative or remnant of notochord that should normally be present. Anything beyond that particular nucleus pulposus, if it is present in terms of notochord, that should be creating some troubles to us in forms of this. So we call this clinical disease as chordoma. You see the lower end of this, just like sacrococcygeal teratoma, we have a tumor growing over here, maybe in the tail end, maybe in this neck end. We call them as sacrococcygeal teratoma or cervical teratoma or cervical chordoma. So this remnant, which normally should not be present, if it is present, creates some diseases in forms of chordoma. So we understand these two phenomena, sacrococcygeal teratoma, which is arising because of the persistence of the primitive streak, and this is known as chordoma, which is appearing because of persistence of excess notochord. Clear or not? So don't get mixed up with this fate or formations of these two tumors, though these tumors are present on the same locations. So apparently it looks like they are same, but they are not. Originally they are from the different sources. One is primitive streak, so you have to know about what is primitive streak and what is persistent, and which is way different from this particular notochord or notochord persistence. So this is kind of thing, and again, these things more commonly is coming in the exam or coming in the future exams will be coming. So more you come with this particular clinical problems with this particular anatomy. Now, if you have observed or looked into this particular previous year's exam, it is all clinical based questions we are going to get. So again, giving this clinical problems, you will be given this particular clinical questions on this embryological development, on this particular anatomical problems. So unless until you will study them well, there's a no cakewalk for you, right? So 1/5 of the students failed last year. You can count it more, like it would be 1/3 for your batch will be failing in the examination because of the poor productions of these answers that you are making. It's not that it's not been covered. It's not you are not practicing and not paying enough attention. That creates the problem. And if you are not paying attention at the right time, you cannot catch up the trend because it's a very fast moving trend. So you will be progressing very fast with the tons of informations. Unless until you digest and absorb through them and repeat them, you're not going to replicate that in during examinations. And apparently, it becomes a very daunting task to cover up everything before examination. You cannot do that because you have a high level of adrenaline which you cannot maintain or which you cannot control over. So that makes these things worse and very worse during the examinations. And many times we see that there are people like writing in a 2 line, 3 lines in a whole page. How can you give marks to it? So in a whole page, if you're writing 3 lines of a clinical problem or any anatomical facts, how do you expect to pass? And that's a fact I am telling it. Even eyes closed, I cannot give marks. For 3 lines, I have to give 30 marks to make you pass. Anyways, so these are the 2 entities that we have mentioned about these particular elements. So forget about the allantois right now. Next big to this particular thing, we call them as new relation or neural tube formation. So our brain or our neural system is nothing but a tube. Now, part of the tube is cylindrical and part of the tube is cylindrical but expanded, which is the cranial end has. So brain part, which is expanded neural tube, which looks like a balloon and it has subdivisions to it. And this particular tube, which is lying in this particular spinal cord, which is lying into this particular vertebral column, is purely tube. Now, this particular tube, which is there, it has openings. So, it has an anterior opening, we call them as anterior neuropore, and it has a posterior opening, which is known as posterior neuro-opening or posterior neuropore. These are the 2 pores. Pores means P-O-R-E is the opening which is present in them. So, these particular entities matter to us. Now, how we are going to make these particular tubes from the flat tissues, just like we have the papers, and from the papers we have to make tubes. So, we cannot make a paper transforms into tube unless until we fold it, and that's what it happens in case of this formation. Now, whatever happens in this particular new relation or neural tube formation is everything triggered by one element. That particular element is the notochord. If we do not acquire the notochord by the gastrulations, we do not have anything further because that's the band master that sets the tune for formation of this. So this is the notochord that is formed under this epiblast layer in the midline. Now this particular notochord transforms this particular epiblast which is lying above to it to transform, to change. It pokes to change chord. So keeps on poking on to change into it. This epiblast which is lying on this particular region, which is ectodermal element, transforms into a new element. We call them this particular element which is known as neuroectoderm. So this neuroectoderm is a kind of modified form of ectoderm which is lying or overlying just above this notochord. Now this particular notochord which stimulates this with some factors changes this particular ectoderm into neuroectoderm. Soon after this neuroectoderm, we have to make this particular flat region into tube. We cannot do it unless until it shows some changes. Soon after that, it makes some groove. So we call them as neural plate groove or neural plate. So this particular groove sets in and elevates the margins of this particular groove as fold. It lifts up on the margins of it. We call them as neural folds. Soon this right side neural fold, left side neural fold meets together and slowly forms this particular tube encircled. And once this particular tube is formed, this particular neural tube get detached from the surface ectoderm or the skin tissue itself. So we have this particular notochord lying in the bottom, then we have the tube, and then we have the skin or epidermis, which is all separate entities and formed over there. This is how this particular tube is forming. Now, while this particular fold is forming, to make this particular tube, a number of the regions, which is the meeting point of this ectoderm and the neuroectoderm, this region, which is dark blue or navy blue, if you see that, this region, which is meeting over here in the medial part, the cells from here starts to migrate out. So this particular migrating cells, which is present in this particular purple, dark blue region, we call them as neural crest cells. They migrate out and separate out themselves from this particular tube and create something else. So this is how this particular entire neural plate, neuroectoderm and neural tube is formed. So this particular tube is formed initially at this particular ectoderm region. We call them as neuroectoderm, as I mentioned, stimulated by the notochord. And this particular thing extends beyond the notochord. That means it's not limited to the notochord only, rather cranially it moves towards this buccopharyngeal membrane and beyond that part as well. And then invaginations and the folding happens. So we have the notochord triggering this particular changes. The neuroectoderm is formed that creates this particular folding inside. We call them as groove. This particular folds moves farther, meets to in the midline, and slowly this particular folds meets in the midline and gets detached from this particular surface ectoderm slowly and forms this particular tube. And this particular entire formations of this particular tube is taking place from the 3rd week to 4th week of life. So this tube is formed by the end of 4th week. So 3rd week on, we have this particular neural tube formed, which is having this particular plate moved into tube, and this is how the folding happens and helps us to form this. Now, as we have the tubes, we have this particular tube is not a solid tube, just like the notochord. This is a hollow tube. Through this particular tube, fluid can pass through, and this particular tube has the 2 ends. One is at the lower end, other is at the other end. We call them as anterior end and the posterior end of the tube. Opening of that, we call them as anterior neuropore and posterior neuropores. So these neuropores matters to us. Clinically, this is very important. Now, this particular neuropores should be closed. So as this tube is formed, the tube need to be closed tube, or else whatever the brain cells are there will be washed out from either to the lower end or the upper end. Many a times we see these particular pores not closing. And if this particular tubes are not closing, the brain material, the neural tissues cannot stay inside. And we have a very grave situation for it. We have the disease arising out of this. So this entire process of formation of the tube and closure of the tube to make it kind of blind tube should be made by the 4th week of life. So if it fails to occur, we have this particular problem. Now, this is what is important to us. As I am saying, this particular folds are fusing to form this particular tubes. So this was a flat thing and it is pouring. So the closure of this particular fold. So we have the entire length of the disc, and not throughout the entire length of the disc, this particular folds or neural folds fuse. So this neural fold fuse at a particular point. Where it does start fusing? It starts fusing in the cervical region. So this particular brain tube which was forming, the closure of the brain tube which is happening by meeting of the folds, that meeting point starts at the cervical region. And this particular cervical closure of the tube happening at the first, and then it zips up and zips down. Am I clear? So we have this particular plate, and this union of the plate is going on to make this particular tube. So this meeting of this particular folds to make this particular tube, that meeting starts at some point. Not the entire tube joins at the same point of time. So this particular meeting of these folds, that's what he is saying here, is taking place in the cervical region, and later on it slowly goes down and up, and this particular anterior cranial pole and the posterior cranial pole are finally fused. So this particular pattern of fusions matters when this particular counting up the age of this particular neural pore formation is concerned. Now, this is what we are talking about, the migratory cells at this folding taking place. We call them as neural crest cells. So, we have these particular yellow dots placed on here. So, these are the region where this particular neuroectoderm and ectoderm is meeting. So, this region is ectoderm and this black region is neuroderm or neuroectoderm. So, this particular meeting point is yellow spots over here. So, this particular yellow spots where this ectoderm meets with ectoderm meets with neuroderm, this particular point, surface point, this region, cells whatever was present becomes a migratory cell. We call them as neural crest cells. So, this particular cells separate themselves and move out like this. So, this is union taking place and this particular element cells moves like this neural crest cell bunch and later on they move out. Now, why do they move out? Now, we have different kinds of neural system present. We have the central nervous system and we have the peripheral nervous system. This particular neuroectoderm migratory cells, which is known as neural crest cells, they give rise to the peripheral nervous system. That means they are going to give rise to the autonomic sympathetic nerves. And from this particular tube, it develops the brain and the spinal cord. So, these are the two different things that happens from this neural crest cell and this neural tube. So, these are the derivatives of the neural crest cells and predominantly it forms this particular ganglion or autonomic nervous system ganglia. So, the autonomic nervous system ganglia in short is contributed by these migratory cells which call them as neural crest cell. Neural crest cells are exam questions. Along with that, this particular neural crest cells transforms into several elements like they forms the adrenal gland medulla. They forms this particular pia mater and aragroid, that meninges layer. They forms this Schwann cell which is present in the neural elements. They forms this thyroid parafollicular C cells. They are neural crest cells. So, you have to know about this particular neural crest cells and where do they come like, where do they come from, how they are formed and what they finally transforms into. Peripheral nervous system predominantly and along with that, these are the contributory elements which is coming from this neural crest cells. So, you have to memorize a couple of them at least for giving a decent answer whenever given answer to it. Now, coming to this neural tube defects, as I am saying, the neural tube, if it fails to fuse, especially this particular entity, we call them as non-fusions of neural pores. That means anterior neuropore, if it is not fused, we do not have any brain formed. All this neural element comes out of this. So, we call them as anencephaly. We do not have any brain formed in this particular problem. Now, why has it happened? It happens failure to fuse the anterior neuropore. Similar to that, if you have non-fusion of posterior neuropore, we get a series of disease, we call them as spina bifida. What is spina bifida? We have this particular spinal cord, spinal meninges or the parts of vertebra which is defective and resulting in this particular spinal cord or spinal nerve or spinal meninges popping up from the back or like this. So, we call them as meningocele, meningomyelocele. These are clustered in a bigger group, we call them as spina bifida disease. So, spina bifida is a disease into which we have 4 different types. If you cannot memorize these types of this at this level, you do not have to, you just have to remember this name spina bifida is a disease which is arised, one of the reasons for it because of this posterior neuropore is not closed. And if this anterior neuropore, if it is not closed, we have a disease, we call them as anencephaly. We do not have any brain tissue. So, this is in short about this particular element of neural tube formations and neural tube formations error. So, you have to prepare this neural tube, how it is formed. You have to prepare this particular element, we call them as neural crest cells. They are kind of exam important areas that you have to prepare. Along with that, we have to prepare the gastrulations and the primitive streaks. Understanding the clinical fate of non-disappearance of this particular element. So, if this particular element, if not regressed, what diseases outcome comes to us to this particular individual, right? So, this covers this particular 3rd week We are left with only few topics in this particular general embryology, and we are ready for exam.
So what is the lining epithelium in the tongue? We have got non-keratinized stratified squamous epithelium, তাই তো? Non-keratinized stratified squamous epithelium. So next কি পাবো? Which organ is there? After tongue, oral cavity, right? After that you are getting the esophagus. So have you studied the esophagus? Histology? No. So next organ is the stomach. Okay. So would be writing this thing about the stomach. So for knowing the whole GI tract, you have to know what are the 4 layers. Okay. From the lumen, GI lumen, throughout it is a pipe, right? So from inside to outside, what are the 4 layers? Okay. So write down the normal 4 layers of GI tract are from inwards to outwards. Okay, from inwards to outwards, number 1 is কি পাবো সবার আগে? Epithelium, right? Number 1 is the epithelium. Now tell me which type of epithelium you will get. So what is the function of stomach? Tell me the extension. Stomach extend from the esophagus up to the small intestine. Small intestine first part ki? Small intestine got how many parts? Duodenum, jejunum and ileum. Okay, so it is extend from the esophagus, stomach, then the duodenum. Okay, so about the stomach, write it down. Epithelium consists of simple columnar epithelium. Simple columnar epithelium. So wherever we got the columnar epithelium, what is the main action over there? Key action পেয়েছি আমরা? Columnar epithelium. So one is absorption, আরেকটা দুটো আসে তাই না? একটা হচ্ছে তোর absorption আর একটা হচ্ছে secretion, right? Okay. So what is the definition of stomach? So if somebody will ask you what is the definition of stomach, what you have to say? Stomach is a muscular bag. Okay. So it is actually receiver of the food bolus, whatever coming from the esophagus. Okay. Then what is happening in the stomach? There is completely mixing and the churning of the food particles. Okay. So this is the main function of stomach. And you know there are numerous glands in the stomach, right? How many glands are there? There are parietal cells, there are oxyntic cells. So what does it secrete? Parietal or oxyntic cell? HCl. So that is the acid chyme, right? So rather than that, what are the other gland? Gastric gland is there, which will secrete which hormone? Gastrin. Okay. And another one is peptic gland is there. So what it will secrete? Pepsin. Okay. Any other gland? Ar kono gland ache? Mucus secreting gland. So this is very much important. Okay. So as because there is numerous acid chyme, it has to be neutralized. তাই তো neutralize হতে হবে। কে আছে তার জন্য? Mucus. Okay. Because afterwards again it will neutralize by the duodenum. You know it will secrete the bicarbonate. Okay. This acid chyme would be neutralized by the bicarbonate. Okay. So write it down in the stomach. You could also write it acidify and converts the ingested food particle into thick viscous pulp. Okay, that is known as chyme. So what is the definition of chyme? Chyme definition ki? It acidifies and converts the ingested food particle. Whatever you are getting, whatever you are taking, whatever it is acidic or basic, doesn't matter, but it will convert, it will acidify and converts the food particle into a pulp-like material. This is known as chyme. Okay. And it also absorbs the water. এখানে কিন্তু একটা ভীষণ ইম্পর্টেন্ট জিনিস, স্টমাক থেকে কি কি জিনিস অ্যাবজর্ব হয় এটা এমসিকিউ আসে। ওকে, নাম্বার 1 is salt. Okay, number 2 is water. Number 3 is drug and number 4 is electrolyte. Okay, these are the 4 things that would be absorbed from the stomach: water, salt, alcohol and also certain drug. ঠিক আছে, So for both male and female should be very much careful about stomach, okay, because two things that will insult the parenchyma of stomach. One is alcohol, okay. If somebody consume too much more, what will happen? The lesser curvature would be, there would be ulcer, okay. Even there would be carcinoma, cancer of the stomach. And for female, what they have to worry? The tamarind water, okay, in pani puri, okay, whatever. In for boys also I am saying, actually it is very much harmful for the stomach, okay, because basically the lesser curvature, okay, ulcer is very common, okay. So these are the things that would be absorbed from the stomach, okay. So see, this is the layer. এবার যদি anatomical gross anatomical structure দেখি anatomy, so কি কি পাবো? First one, this is the esophagus. এই এন্ডটাকে কি বলে? কার্ডিয়া। কেন বলছি? because it is very much proximal to the heart, okay, so that's why it is called cardia. Next one, this one is the fundus, okay, this part is the fundus, this is the body of the stomach. And see, in the stomach there are three type of muscle. কোথায় কোথায় three type of muscle পাবে? দুটো জায়গায়, একটা হচ্ছে stomach আর একটা হচ্ছে urinary bladder, ঠিক আছে। এখানে তিনটে লেয়ার কি কি আছে? আউটার লংগিচুডিনাল, মিডল সার্কুলার এন্ড ইনার অবলিক, ওকে। এন্ড ইন কেস অফ ইউরিনারি ব্লাডার কি পাবে? সেম, আউটার লংগিচুডিনাল, ইনার সার্কুলার, আর মোস্ট ইনসাইড কি মাসল থাকে? ডেট্রুসার মাসল, ঠিক আছে। ইউরিনারি ব্লাডারে ডেট্রুসার মাসল থাকে। সো হিয়ার উই আর হ্যাভিং থ্রি লেয়ার্স, okay, and see, this is the lesser curvature and this is the greater curvature. So that's what I'm ulcer of the stomach is common in the lesser curvature, remember this one, okay. And if carcinoma, it is common in the greater curvature. কেন greater curvature বেশি হয়? because এখানে প্রচুর পরিমাণে lymph nodes থাকে, okay. There are 5 type of lymph nodes in the stomach, so they are present on the lesser curvature. A greater curvature is common for the ulcer. Now coming to the histology, okay. So histology, what are the 4 basic layer you will get? First one is the epithelium, okay, then the mucosa, okay, then the muscularis externa, and the last one is the serosa, okay. So first you are getting the epithelium, okay. Epithelium, you know, simple columnar epithelium. আরেকটা কি দেখা যায়? There are numerous gastric pits. This is the identifying feature for stomach, okay. There are presence of এগুলো কি? These are the gastric pits, okay, found on the mucosa of the stomach, okay. So other thing you have seen yesterday on the dissection, right? Inside of the stomach there are the muscle of the stomach having numerous folds, right? These are known as rugi, okay, exactly rugi. বাংলায় যেটা আমরা রুগী বলি, not ruge. Somebody is saying yes, it is rugi, okay. And mucosa also shows tiny grooves, এটা লিখে রাখ, okay. Mucosa not only having rugi, also having tiny grooves, ছোট ছোট দেখবে grooves ওর মধ্যে তৈরি হয়েছে, okay. So these are known as gastric pits or foveolae gastricae, foveolae gastricae. Mucosa also shows tiny grooves which appear as invagination. These are known as gastric pits or foveolae gastricae, foveolae gastricae. কিছু কিছু রুগী দেখবি ইনসাইডে ওটা আরো যেন ভেতরে যাচ্ছে, রাইট? ইট ইজ মেকিং সাম কাইন্ড অফ চ্যানেলস, সো সাম কাইন্ড অফ পিটস, more deeper pits. So these are the foveolae gastricae, okay. So see, this is the rugi, right? And what is this? Pyloric sphincter. So একটা কি হয়? You have to know the anatomical position of stomach, ঠিক আছে? কি করে বুঝবি? ধর যদি কেউ স্টমাকটাকে এইদিকে করে দেয়, then also it is having both end. So how to identify যে কোনটা কার্ডিয়াক এন্ড কোনটা পাইলোরিক এন্ড, কি করে বুঝবি? So you have to feel কার্ডিয়াক এন্ডটা কি হবে? কার্ডিয়াক এন্ড would be smooth, okay, but এখানে যদি পাইলোরিক এন্ড পাস, it would be more thicker, it would be swelled up, okay. এই এন্ডটা কিরকম হবে? সোয়েলিং থাকবে, so that is known as pylorus or pyloric sphincter, okay. So দুটো এন্ডকে আমরা ডিফারেনশিয়েট করতে পারবো। and always লেসার কার্ভেচারটা কি থাকবে? ইনসাইড, গ্রেটার কার্ভেচারটা আউটসাইড থাকবে। বাট সি দ্য শেপ অফ দি স্টমাক ইজ ভ্যারি ইন ডিফারেন্ট ইন্ডিভিজুয়াল। ওকে, স্টমাক যখন ডিটেলসে পড়বে 4-5 ধরনের শেপ থাকে, ঠিক আছে। সো নেক্সট ওয়ান ইজ all the glands, যা glands এখন অবধি পড়লাম আমরা, অক্সিনটিক, গ্যাস্ট্রিক, পেপটিক, whatever gland is there, it is open on the bottom of the gastric pits. কোথায় ওপেন করে? bottom of the gastric pits. Write down this point. All the glands of stomach open into bottom of the gastric pits. Bottom of the gastric pits. Okay, next point আছে যে what is the differentiation between histological and anatomical stomach. Okay, যখন আমরা anatomical stomach করব there are 4 part, but histological কটা 3 part. anatomical 4 part বল কি কি? এই মাত্র বললাম, কার্ডিয়া, ফান্ডাস, বডি, পাইলোরাস। ওকে, বাট হিস্টোলজিক্যালি কি পাচ্ছি? 3টে পার্ট। কেন পাচ্ছি? বিকজ বডি আর ফান্ডাস এদের হিস্টোলজিক্যাল ফিচার ইজ সেম। Okay, so write down the histology only. Histological part of stomachs are এক নম্বর হবে কার্ডিয়া, সেকেন্ড হবে বডি অ্যান্ড ফান্ডাস, and the third one হবে pylorus. Third one is pylorus. Okay, so these are the 3 division. Now see over here, ekhane if somebody has taken the specimen from here, from the body and fundus, so what are the layer they will get? These 3 layers. Okay, so mainly muscularis externa would be very nicely visible on where? On the body and fundus. Outside longitudinal, middle circular, inside oblique. Okay, now write it down. General features. Mucosa তে কি থাকবে? Mucosa has got 3 layers. Mucosa has got 3 layers. কি কি layers থাকে? প্রথমে হচ্ছে epithelium. Number 1 is epithelium. Second one is lamina propria. আমরা অন্য যে কোনো অর্গানে ল্যামিনা প্রপিয়া কোথায় পেয়েছি? একদম বেসে, রাইট? কিন্তু এখানে কিন্তু এপিথেলিয়ামই পাচ্ছি আমরা, ঠিক আছে। মিউকোসার মধ্যে পাওয়া যাচ্ছে এপিথেলিয়াম, ল্যামিনা প্রপিয়া, দেন মাসকুলারিস মিউকোসা। মিউকোসার মধ্যে একটা মাসেল লেয়ার থাকে যেটাকে বলছে মাসকুলারিস মিউকোসা। ওকে, দেন দ্য সেকেন্ড ওয়ান ইজ সাবমিউকোসা, দেন মাসকুলারিস এক্সটার্না, and last one is serosal layer or adventitial layer. Serosal layer or adventitial layer. Okay, so histological feature you have to say these all 4 points. Okay, among that in the mucosa you have to say epithelium, lamina propria, muscularis mucosa. Okay, so next one, see this is what is happening. First one, you have taken one specimen from here, so it is having, first one is these are the gastric pits. So gastric pits টা কি হচ্ছে? This is elongated structure যেখানে একদম নিচে আমরা দেখতে পাচ্ছি ল্যামিনা প্রপিয়া। ওকে, সো এই লেয়ারটার মধ্যে 3টে আছে, সারফেস এপিথেলিয়াম যেটা হচ্ছে সিম্পল কলমনার এপিথেলিয়াম, then this is the lamina propria and this is the muscularis mucosae, and whatever the glands it will open where? At the bottom of the gastric pits. Okay, then the next layer is the muscle layer. Muscle layer having three layers again, and the last one is serosa. Serosa te ki thakbe? Blood vessels. Okay, numerous arterioles and venules. Okay, present where? In the serosa. Okay, so see the glands. If you are taking the glands, okay, so what is the basic structure of gland? কি কি থাকবে? প্যানেট সেল। প্যানেট সেলটা কোথায় পাওয়া যায়? মেক ওয়ান স্টার মার্ক। প্যানেট সেল ইজ ফাউন্ড at the surface epithelium, at the surface epithelium of gastric glands. Pannate cell will also secrete one type of thing. What is that? Lysozyme. Okay, pannate cell এটা একটু দেখে নিবি। Okay, which is the substance that would be secreted by pannate cell. Okay, then next one is, this is the chief cell. Chief cell or oxyntic cell will secrete HCl. Okay, and then enteroendocrine cell, it will secrete the gastrin and the pepsin. Okay, so write down this thing, mucosa. Just write simple tall columnar epithelium. Just write this one. Okay, this sentence you write, simple tall columnar epithelium which secrete mucus, okay, that lubricates and protect. It has got two function, lubricates and protects. Lubricates and protects the epithelial surface from the acid content. The epithelial surface from the acid content. আচ্ছা, এখানে আরেকটা জিনিস ভীষণ ইম্পর্টেন্ট, the epithelial cell are renewed about every 3 days. So could you imagine প্রতি 3 দিনে কিন্তু এপিথেলিয়ামটা রিনিউ হয়। ওকে, আমাদের স্কিন কতদিন অন্তর রিনিউ হয়? Do you know? না, কাছাকাছি গেছিস। 28 days, মনে থাকবে 28 days। তেমনি স্কিন সেল শেড করে, ঠিক আছে। উপরের যে লেয়ারটা, উপরের লেয়ারটা কি? ডার্মিসে কটা লেয়ার? 3টে। কি কি? হ্যাঁ, একটা এপিডার্মিস, হাইপোডার্মিস এন্ড এন্ডোডার্মিস। সো এপিডার্মিসটাই কিন্তু শেড করে, উপরের যে লেয়ারটা সেটা শেড করে, সেটা কখন করে? 28 ডেজ, এটা মনে রাখবে। ওকে, বাট এখানে দেখছি কখন এপিথেলিয়াল সেলটা স্টমাকে রিনিউ হচ্ছে এভরি 3 ডেজ। ওকে, সো সি দিস ইজ দ্য স্ট্রাকচার, this thing we have already described. So now mucosa lamina propria te ki thake? Okay, write down lamina propria contains gastric gland. So there are 3 type of gland, either it would be cardiac, it would be fundic or pyloric. Cardiac, fundic and pyloric, these are the 3 type of glands. Okay, then muscularis mucosa. So muscularis mucosa contain, I told already there would be some muscles, but which type of muscle? Ki thakbe? Smooth muscle, remember this one. Okay, so muscularis mucosa made of 2 layers of smooth muscle. Okay, smooth muscle fiber extend into lamina propria between the gastric gland. এই smooth muscle টা এমনই থাকে, সেটা কি করে? lamina propria যেখানে gastric gland থাকে, it will invaginate between that. Okay, so actually it is supporting that one, supporting the structure. So that is about the first one, mucosa. Okay, so see, this is the gastric pits, it is completely elongated thing. Okay, and you could see these are the gland. Okay, now the submucosa. The submucosa is a thick layer. Submucosa te ki thakbe? This is nothing but the connective tissue layer. Okay, submucosa is nothing but the connective tissue layer. যখনই তোরা histological feature লিখবি, just write down one line. Okay, so about submucosa, about mucosa. Mucosa you have to elaborate the 3 things. Then submucosa you just write it is the connective tissue layer. Okay, connective tissue layer that surround the mucosa. So connective tissue থাকলে কি থাকবে এখানে? Blood vessels থাকবে, lymphatic থাকবে and the glands থাকবে। Blood vessel, lymphatic and the glands. So which type of plexus is found there? Do you know? না, GIT তে 2 ধরনের লেয়ার থাকে না, একটা তোর আয়ুর্বেদ প্লেক্সাস আর একটা হচ্ছে মাইন্টারিক প্লেক্সাস। তো এখানে কি পাবো? আয়ুর্বেদটা এখানে পাবো। ওকে, মাইন্টারিকটা কোথায় পাবো? মাসেলে, লাস্ট লেয়ারে। ওকে, হ্যাঁ হ্যাঁ হ্যাঁ, that is another name is Messinger plexus. Okay, it has got numerous name. ঠিক আছে, আয়ুর্বেদ or Messinger plexus আর ওখানে থাকবে মাইন্টারিক plexus. Okay, so these are the plexus of the vessels. Okay, now see each and every cell they have described. So you just write it down, how many type of gastric glands are there. Okay, কি কি টাইপ আছে? Mucus neck cell which will secrete the mucus, chief cell, parietal cell, endocrine cell. So what is the difference between this chief cell and parietal cell? দুটো কিন্তু আলাদা করেছে এখানে। এখানে একটা ফ্যাক্টর কি ইয়ে হয় না? হ্যাঁ, সেটা কোথায় পাওয়া যায়? প্যারাইটাল সেল থেকে হয় না, বাট চিফ সেল থেকে কি হলো? HCl। তার মানে এটা কিন্তু দুটো আলাদা আলাদা সাবস্টেন্স সিক্রিট করছে। ওকে ওকে, সাবমিউকোসা উই হ্যাভ, সো ইউ আর কারেক্ট, দিস ইজ মেইসিনার প্লেক্সাস। ঠিক আছে, লিখবি? হ্যাঁ, যাদের যা ট্রেন আছে চলে যাও। এই ক্লাসটাতে ওটাই সমস্যা, সবাই ট্রেন। শনিবার লাস্ট ক্লাস, কটা বাজে? না, যাদের ক্লাস আছে, এই যাদের ট্রেন আছে তারা বেরিয়ে যাক, ঠিক আছে, কোনো প্রবলেম নেই। ওকে, সো here it contains numerous blood vessels, lymphatics and Meissner plexus. Okay, so this is the gastric pits. Okay, now next one is muscularis externa. Okay, so write down muscularis externa having 3 layers. Muscularis externa having 3 layers: oblique layer, circular layer and the longitudinal layer. Okay, oblique layer, circular layer and the longitudinal layer. This thing already I have described. Okay, now between the two layer there is Ayurveda or mesenteric plexus. Oh, actually mesenteric plexus is present in the muscularis mucosa, but here the mesenteric plexus, another name is Ayurveda plexus, both are same. Okay, so that is present where the muscularis externa. Okay, so this is little bit confusing. Mesenteric plexus টা কোথায় পাওয়া যাবে? সাবমিউকোসাতে, কিন্তু muscularis externa তে পাওয়া যাবে Ayurveda or mesenteric. These two is the same. Okay, and the inner circular layer act as a sphincter. Which sphincter? That is the pyloric sphincter. Okay, so you already know physiological sphincter পড়েছিস, ওটা anatomical sphincter। কার্ডিওইসোফেজিয়াল জাংশনটা পড়েছিস স্ফিংক্টারটা ফিজিওলজিতে? হ্যাঁ, কার্ডিওইসোফেজিয়াল জাংশন মানে যেখানে তোর ইসোফেগাস আর স্টমাকটা কানেক্ট হচ্ছে, ওখানে একটা স্ফিংক্টার থাকে, দ্যাট ইজ নোন অ্যাজ lower esophageal sphincter, or this is also known as cardiac sphincter. It is not a true anatomical sphincter, মানে অ্যানাটমিক্যালি এর কোনো অস্তিত্ব নেই। ওকে, কিন্তু ফিজিওলজিক্যালি আছে। ওকে, রিমেম্বার দিস ওয়ান। বাট ট্রু স্ফিংক্টার কোনটা? পাইলোরিক স্ফিংক্টার। অ্যানাটমিক্যালি তুই স্ফিংক্টারটা দেখতে পাবি যখন অ্যানাটমিক্যালি পাইলোরাসটাকে ক্রস সেকশন করবি, সো ইউ উইল গেট টু নো হুইচ ইজ পাইলোরিক স্ফিংক্টার। ইট ইজ এ ট্রু অ্যানাটমিক্যাল স্ফিংক্টার। বুঝতে পারলি? So this is the numerous layers in the stomach. Okay, now last layer is the serosa or adventitial layer. Serosa or adventitial layer, so it is merely visceral peritoneum. This is nothing but the visceral peritoneum. Peritoneum মানে কি? whole GI tract covering, তাই তো, তাকে কি বলে? peritoneum বলে। So peritoneum are also having 2 layer, একটা parietal layer আর একটা visceral layer. Visceral layer টাই actually serosa. Okay, and serosal layer তার মানে it will epithelium কি হবে? Simple squamous epithelium. Remember this one. The epithelium for serosa in the stomach is simple squamous epithelium. আচ্ছা, এখানে একটা জিনিস, the esophagus lies outside the abdominal cavity, তাই তো, ডায়াফ্রামের উপরে থাকে, অ্যাবডোমিনাল ক্যাভিটির বাইরে। হ্যাজ এ সুপারফিশিয়াল লেয়ার কল্ড অ্যাডভান্টেশিয়া হুইচ কনসিস্ট অফ অ্যারিওলার কানেকটিভ টিস্যু। এটা ইসোফেগাস সম্বন্ধে বলেছে, ঠিক আছে, যে ইসোফেগাসের লেয়ারটায় কি থাকে তাহলে? অ্যারিওলার কানেকটিভ টিস্যু। অ্যারিওলা আছে মানে কি? ফ্যাট আছে, ফ্যাট সেলস আছে। ওকে, সো দিস ইজ অ্যাবাউট দ্য সেরোসা। তাহলে হিস্টোলজিক্যাল স্লাইডটা এরকম খানিকটা দেখতে হবে, ঠিক আছে। গ্যাস্ট্রিক গ্ল্যান্ড হবে, গ্যাস্ট্রিক পিটস থাকবে, and this is simple columnar epithelium, lamina propria and the gastric pits. Okay, so see, you could take the snapshot. Okay, these are the type of cells. Okay, এখানে কোনগুলোকে, these are the gastric pits, যেগুলো longitudinal আছে সেটা হচ্ছে gastric pits, কিন্তু wherever you are getting this small foramina, that is known as gastric foveolae or foveolae gastricae, something we studied this one. And these are, এই যে মাঝখানের লেয়ারটা, this whole big layer, this is nothing but the numerous gastric gland. Okay, and at the lower layer again you are having this muscularis mucosa. Okay, and here muscularis mucosa নিচে কি থাকবে, সাবমিউকোসা, তারপরে কি থাকবে, muscularis externa থাকবে। ওকে, আর লাস্ট লেয়ারে কে থাকবে, দ্যাট ইজ দ্য সেরোসা। ওকে, সো সি এখানে খুব ভালোভাবে বোঝা যাচ্ছে। তাহলে প্রথমে গ্যাস্ট্রিক পিটস, তারপর নেক্সট তুই কি পেলি, পুরো হচ্ছে গ্যাস্ট্রিক গ্ল্যান্ড। ওকে, ইট উইল অকুপাই দ্য ম্যাক্সিমাম সারফেস এরিয়া। দেন নেক্সট ওয়ান ইজ, দিস ইজ দ্য মাসকুলারিস মিউকোসে। সো এখানে একটা বর্ডার লাইন থাকবে, অ্যাজ বিকজ একটু স্মুথ মাসেল আছে, সো দ্যাটস ওয়াই ইট ইজ ক্রিয়েটিং হোয়াট, দ্য বর্ডার, lower border, so that is muscularis mucosa and this is the submucosa. Okay, এখানেও ওই অবধি আছে, তারপরে নিচে আরেকটা লেয়ার যদি দেখতে পাস সেটা হচ্ছে সেরোসা। ওকে, এটা লেবেল নেই, তাহলে বল এটা কি, না এটা কি, এগুলো গ্যাস্ট্রিক পিটস। আচ্ছা এইগুলো কি গ্যাস্ট্রিক, এইগুলো গ্যাস্ট্রিক গ্ল্যান্ড হবে। এটা কি, ওই যেটা ফোভিওলে গ্যাস্ট্রিকে যেটা বললো, বিকজ দিস ইজ রাউন্ড, ঠিক আছে, এগুলো রাউন্ড হলো, এইগুলো কিন্তু গ্ল্যান্ড। ওকে, দেন এই লেয়ারটা কি, মাসকুলারিস মিউকোসা, এই লেয়ারটা সাবমিউকোসা, তারপরে এটা হচ্ছে সেরোসা। ঠিক আছে, সো দিস ইজ দ্য লেবেল ডায়াগ্রাম। ওকে, সো জাস্ট গো অ্যান্ড সি দ্য স্লাইড। ওকে, so that is conclude about the histology of stomach.